Hello students, today we will discuss about gas chromatography. Gas chromatography actually can be used to separate volatile compounds. What is volatile compounds? The compounds which can convert into gas very quickly. Okay, so it is the whole setup of your gas chromatography. Here one oven containing coiled column is used, then one sample injector or injector oven with a very high temperature, because it will convert all the samples into gaseous form. Then through this injector we can also inject our carrier gas or mobile phase. Then the sample will enter through this and it will move, and finally waste gases released through this chamber or canal, and finally we can analyze it through mass spectrometer. These graphs, we obtain these graphs. From this graph we can predict different gases. So inside that coiled column our stationary phase is present. What is our stationary phase for gas chromatography? It is a liquid with a very high boiling point, because inside the oven the temperature varies between 50 to 250 degrees Celsius, which is very high. So we can't use a liquid with very low temperature, because if we use one liquid with very low temperature, it will convert into gas very quickly. So we use a liquid with very high temperature, high boiling point. For example, waxy polymer. Okay, that liquid is adsorbed on a solid, generally which is diatomaceous earth. What is adsorption? The liquid molecule will attach on the surface of the stationary phase or a solid, which is diatomaceous earth. Then what is the mobile phase here? The mobile phase is an inert gas which will not react with any molecule here. So generally we took helium. Okay, then we will inject our sample through some injector oven. So rate of the sample movement through this coiled column depend on two factors. One is boiling point of the sample. Sample with high boiling point will release later on, but sample with low boiling point will release fast. So sample with high boiling point can interact more with the stationary phase, so it will release slowly and later on, but sample with low boiling point can move faster in a faster rate and release at first. Then the interaction with stationary phase, the gas or the sample which interact more with the stationary phase will release later on, but the sample which interact less with the stationary phase, it will release fast. Okay, so if we increase the oven temperature, then gases will release in a very faster rate. So we can't analyze those, calculate those graphs, those peaks of different components of gases in this faster rate. But if we lower the oven's temperature, gases will take some time and release slowly, so that we can analyze those carefully and systematically and accurately. Okay, so this is all about your gas chromatography. I hope you really enjoyed this, will get benefit from this video. So if you liked it, please hit the like button, please share with your other friends and family members, and please subscribe to my channel to get more important videos like this. Thank you.